Hello and welcome, dear friends, to this concept of the Lac Operon. In the previous lecture, I had discussed about the operon system, gene, what is the function between a DNA and a chromosome, and some aspects of regulatory mechanisms of operon. So before actually venturing into deeper concepts of gene regulation, I would like to give you two examples in two separate lectures of the operon systems in E. coli. One is the Lac Operon, which is a catabolic type of mechanism, and another is tryptophan operon, which is an anabolic type of mechanism. But what does it mean when I say a catabolic operon and an anabolic operon? For that, you have to understand that whenever we talk about operon system, we are talking about a gene expression, and gene expression is nothing but protein synthesis. So when you are dealing with protein synthesis, you are dealing with transcription and translation from the DNA. That is, in between the protein and the DNA, there is a mRNA. These are the key points where the gene gets regulated. What is regulation? Regulation is putting the protein synthesis on when needed and putting it off when not needed. So in simple terms, gene regulation or operon function is meant for putting the genes on and putting the genes off. How exactly does the cell know when to put the gene system on, means when to produce a protein and when not to produce a protein? That is dependent upon the stimuli or the stimulus which the cell gets in the presence of a substrate or in the lacking of a substrate which is needed for growth. So for that, if you just take a look into the definition of metabolism, that is catabolic pathways and anabolic pathways. You see, catabolism is breakdown of larger molecules into smaller molecules. That is, for example, carbon substrates like lactose, sucrose, glucose. These are broken down into smaller molecules from which energy is derived. That energy is invested in creating cellular macromolecules like amino acids, proteins, hormones, maybe cell components like flagella, cell wall, etc. So synthesis of larger molecules which forms the component of a cell, part of a biomass, is called as anabolism. And for this purpose, the carbon source which is utilized and broken down to derive energy, that is catabolism. Now, so see, in simpler terms, catabolism is large molecules broken down into small molecules, and anabolism is small molecules assembled to form large molecules. Best example in catabolism, this large molecule is lactose in E. coli, broken down to small simpler sugars, monosaccharides, disaccharides, etc., and that energy used for energy production, and energy is used for anabolic reactions. So we will be studying tryptophan operon as an anabolic mechanism and lactose operon as a catabolic mechanism. So in anabolism, larger molecules which forms the part of the cell, that is cytoplasm, cell wall, ribosomes, by the mechanism of formation of proteins, etc., is taking place. And here we will take tryptophan synthesis, amino acid being synthesized when not available, as an example in the next lecture. Today's lecture we will be dealing with lactose utilization, where the nutrient in the form of lactose is utilized, broken down into smaller molecules and derived for energy production, which is used for anabolic reactions. So lactose is a carbon source and Lac Operon is a catabolic operon, where these nutrients, if you see here, let us consider this as lactose. There is a cell wall or a barrier between the environment where this carbon source is present and the cytoplasm where it needs to be transported. So uptake of lactose is the key thing, the very prime thing which the cell has to do, and for that purpose it has to secrete certain enzymes through the pores of the cell wall, act on the carbon source, that is lactose, and take it inside. Now for this you may be wondering that this is an example of transport. Yes, actually it is an example of transport. We have two types of transport, active transport and passive transport. Remember here one thing, passively some amount of lactose goes inside in the very beginning. There may be huge amount of lactose present as a carbon source in the medium. Example is a Escherichia coli cell growing in a McConkey's broth or a McConkey's agar. What is there in the agar or the broth? Huge amount of lactose. The cell has to take that lactose inside, and for that purpose it has to secrete certain enzymes. That enzymes need to be activated by lactose itself, and this whole beautiful mechanism is initiated by initially a passive diffusion of this lactose molecules inside, which then activates this Lac Operon. This activation results into certain enzymes, where these enzymes are uptaking the carbon molecules inside, breaking down into simpler molecules and utilizing it for the purpose of energy. So in this lecture, our whole purpose is to see that gene on the DNA which is responsible for secreting those enzymes which will utilize lactose. Like this, hundreds and thousands of operons may be existing in the cell. We are taking Lac Operon as a basic example, a very beautiful example, which is a representative of all the operons in the cell. Secondly, in my next lecture, I will talk about tryptophan operon, which is a anabolic operon. But let's just see why lactose operon is a catabolic operon. Here at this juncture, you should understand, you should bring in your knowledge about genetics, enzymology, physiology, metabolism, all combined here. Means this operon system can be seen through a metabolism perspective, seen through a genetics perspective, or can be seen through a enzymology perspective. It's up to you how you take out the information and knowledge from this. So let me tell you once again, this lactose nutrient, consider this as lactose, will be taken inside. It will be broken down to glucose, actually broken down to beta galactosidase, beta galactose, and then to glucose ultimately. During this process, some enzymes are needed. These enzymes are proteins, and these proteins have to be regulated, synthesized from DNA. That segment of the DNA will be the center stage of discussion here, and that segment of DNA is nothing but called as the Lac Operon, which will be made up of various other parts. But before going directly into the theoretical aspects of Lac Operon, I would like to show you this whole concept in animated fashion. A small animation and along with it some explanation, and then we will come back to the PowerPoint slides. I hope you enjoy the animation as well as you understand and extract knowledge from it. Come, let's see the animation first. So let us try to understand this Lac Operon in three parts. The first part is utilization of lactose when lactose is present in the media and how it is transported inside and taken up for degradation. First thing. The second aspect of seeing the Lac Operon is seeing it as an inducible system, where the lactose itself induces its own degradation. Means as long as lactose is present in the media, the operon will be on, and when lactose is over or when there is no lactose in the environment, this operon will be shut off. Means lactose itself induces its own biodegradation, so it is an inducible operon. Second aspect. The third area which you have to understand is when lactose and glucose both are provided in the media, it is very interesting to know that the cell will utilize only glucose. Means if lactose and glucose is present, only glucose will be utilized, not lactose. In that case, lactose has to be put off. The Lac Operon has to be put off. So there is a cyclic AMP and CAP combination of function which is called as an activator protein. So we will see this in three parts. So glucose, you should understand, is the main food source which is used by bacterium, as it does not need to be modified, one thing. And many enzymes are required for lactose metabolism, which makes it inefficient to metabolize lactose when glucose is present. Means why to synthesize so many enzymes for utilization when glucose can be directly taken and put it into a pathway. So when glucose and lactose both are present, try to understand bacteria, it will favor the glucose metabolism over lactose. In absence of glucose, bacteria will turn on genes necessary for lactose utilization, and that is what we are going to see. So the Lac Operon is one of the best understood examples of the control of gene expression, and it was first discovered by Jacob and Monod, and this was given the name Lac Operon. It is a operon. It is a genetic unit which consists of one or more structural genes and adjacent operator genes. Means there are two parts in this operon. One is regulatory gene, other is structural genes. Structural genes are the main genes which will be responsible for enzyme synthesis. So we will see this. This is a operon. So I, P, O, Z, Y and A. These are the operon system, operon parts of the Lac Operon. So it is negatively controlled in this example, as you can see here. So this I forms a part of regulatory genes, I, P and O. These are the regulatory genes. Z, Y and A, these are the structural genes. Structural genes means which are actually responsible for the proteins, that is the enzymes. Three enzymes will be present here. Glucose is present, lactose is absent, repressor binds to operator, blocking the transcription process. So in response to the environmental signal, certain regulatory proteins are made. See, there are two types of proteins made. One which are called as regulatory proteins, the other which are called as the structural proteins. So regulatory proteins, they mediate the gene regulation by increasing or decreasing the regulation of these structural genes. Means these regulatory genes are responsible for controlling these structural genes. Okay, so this can be in a negative fashion, that is a negative control, or a positive control. Lac Operon is said to be under negative control because there is a repressor formed. This blue molecule, it is a repressor formed, and this repressor blocks the operator. You see, I is a regulatory gene which forms a repressor. This repressor, it blocks the operator site. P is the promoter site for binding up of polymerase. This is polymerase, RNA polymerase. This whole segment is DNA. Be very careful in listening. This is DNA on which promoter is there. This promoter binds RNA polymerase. RNA polymerase will be responsible for mRNA, which will be translated into three enzymes, Z, Y and A. I produces a protein which is not a structural protein, but it is a repressor protein, which always means, let us say, consider in a cell where there is no lactose. Always lactose may be present, may not be present, but what is there always on the gene, always a repressor is produced and it is blocked at the site of operator. Means 24 hours, whenever you see inside a cell, this repressor is always bind to the operator. So at any given instance of time, in Escherichia coli, you will find that this repressor is always bound to the operator, blocking the path of RNA polymerase. How will this repressor be removed? This will be removed if lactose is present in the media. So lactose induces the removal of this repressor. Therefore, Lac Operon is always called as an inducible system. Why it is called as an inducible system? Because lactose in the media will come and remove this repressor and start the synthesis of structural genes. So Lac Operon is always called as an inducible system. It is always under a negative control because this is repressor always blocking the operator. So basically Lac Operon is negative controlled, but it can be positive controlled also. In an example of positive control, there is a CAP-CAMP system, which I will explain later on. There is a CAP-CAMP system, a complex which bind to the CAP site on this operon. Where that is, I will show you. Binds to the CAP site and it facilitates the RNA polymerase binding at the promoter. Means for binding of this RNA polymerase at the promoter, there is a need for some activator protein. And so the transcription will be on. So that I will discuss in detail later on. That is the positive control, which involves the cap binding site, CAMP and the CAP. We will see it afterwards. But first let us move on to the negative control. And also understand that the cis and trans functioning of Lac Operon exists. This I have explained in my last lecture. Genes, they have the ability to affect other genes. Means this regulatory genes, it affects the structural genes. Regulatory genes, it controls the structural genes. So just like in a higher chromosome, there is a cis and trans dominance. Cis means adjacent and trans means across. Cis means adjacent to the structural genes. Cis genes are the regulatory genes, and trans genes are those genes which are actually involved in the protein production. See, trans, and trans, this I is also called as trans because that is involved in a protein production, whereas P and O, they do not form protein but they attach protein to themselves. So if a DNA sequence can have an effect over another DNA from a distance, means this I which is away from Z, Y and A, but still it affects Z, Y and A being away from it, then it is called as a trans acting site. Okay, and a cis acting is the one which does not form a protein, but the DNA is near to it and it is attaching certain proteins to the DNA. So these are the regulatory sites. Regulatory sites are genes which control the production of proteins. These DNA sites can act on adjacent genes on the same piece of DNA, regulatory DNA away from the gene cluster but still affecting the structural genes. In Lac Operon, Lac O and P, these are cis genes because they do not produce diffusible protein. They act on the same DNA molecule, that is which produces I, Z, Y and A, and these are structural genes, that is I, Z, Y and A. They produce diffusible proteins, therefore they are called as trans genes. So cis and trans, this is called as a cistron, and the cistron is a cluster of genes which produces more than one type of enzymes, then it is called as a polycistron. If this cluster of gene is responsible for, which seldomly happens in bacteria, but if it is responsible for production of only one enzyme, then it will be called as a monocistron. So cis and trans dominance is there in Lac Operon, and that is what the regulatory mechanism, that is the combined function of genetic system, is all about. So you see this gene cluster I, which produces the repressor protein. The repressor gene, it produces the repressor protein and it regulates the transcription by binding it to the operator gene. It is not part of the Lac Operon, but it is a region which controls the production of structural genes. P and O, these are the promoter region and the operator region. Promoter, you know, is the binding site for RNA polymerase, and operator is the site for binding up of the repressor produced by this I gene. The operator has a binding site for repressor produced by the I gene. Lac Z gene is a gene which encodes for beta galactosidase, enzyme which converts the disaccharide lactose into monosaccharides glucose and galactose. Now this is a very important gene, Lac Y, that is it encodes for beta galactoside permease. Now you see, if this, even if this is the last gene in the cluster, but it is the first enzyme needed to permeate the lactose molecule inside the cell. Last gene in the cluster but first to be needed. How is this induced? This is induced by the passive diffusion of certain lactose molecules which move inside the cell. Initially, some molecules which passively come inside, they activate this gene and they start producing this permease, and permease brings larger molecules of glucose inside. So Lac Y gene codes for beta galactoside permease, which will assist the transport of lactose inside the cell. Now there is one more gene called as Lac A, which encodes for trans-acetylase. Now we will not discuss much about trans-acetylase because the actual role of trans-acetylase gene in Lac Operon is not known. Maybe that is in sequence, evolution has maintained in sequence for some other metabolic activity which is related to lactose. So we will not discuss this. We will discuss only three: I gene, which is a repressor; P and O gene, which are the regulatory genes, promoter and operator; Z for beta galactosidase, and Y for permease. So Lac Operon can be defined as a group of genes which ultimately produces the enzymes needed for lactose metabolism. Lac Operon is made up of 5 parts, 3 structural genes, 1, 2 and 3, IPO, 3 structural genes, made up of 3 structural genes, Lac Z, Y and A, 3 structural genes, I repeat, Z, Y and A, and 2 structural genes, P and O. Promoter and operator, it receives the RNA polymerase. It is a template for binding up of the RNA polymerase to initiate the transcription. mRNA will be formed. Okay. So all this is needed to break down the lactose molecule into glucose and water. The enzyme beta galactosidase will break this bond into two glucose molecules. So see here, the enzyme is produced by Lac Z, that is beta galactosidase, which converts this into glucose and galactose. The enzyme converts disaccharide lactose into monosaccharides galactose and glucose. Glucose will be sent to EMP pathway. Galactose by some gal operon will be converted into glucose and then again sent into the EMP pathway. So let us see the operon model. In this diagram, in this animation, you can see the operon contains of structural genes Z, Y and A, and as well as promoter and operator. The Lac I gene, the far left here, produces a protein called as a repressor protein. This repressor protein has an operator binding site and a repressor lactose binding site. This is the lactose molecule. If this lactose binds here to the repressor, it will inactivate the repressor. If lactose is present, now see the chronology or see the narrative here. If lactose is present, operon should be on. If lactose is not present, operon should be off. So if lactose is present, the repressor binds to the lactose molecule and transcription is taking place. Transcription proceeds. Okay, ultimately producing the enzymes to metabolize the lactose. If lactose is not present, the repressor binds to the operator, transcription will not take place. Simple. And this type of regulation is called as a negative control because the transcription only occurs when the repressor does not bind to the operator. So all the time, most of the times, always in the cell, repressor is actually bound to the operator. Only when lactose will be added, it will bind to the lactose molecule will bind to the repressor and then it will remove the repressor. When lactose is not present, it is efficient. When lactose is not present, no lactose, you can see lactose bind allows the binding of repressor to operator. If lactose was present, this repressor would not have bound to the operator, and this polymerase is not functioning because polymerase path is blocked by the repressor. So when lactose is not present, remember it is energy efficient. It is efficient not to produce the enzymes for lactose metabolism. Operon should be off. As there is no lactose for the repressor protein to bind to the repressor binds to the operator. Okay, RNA polymerase tries to bind to the promoter. It may bind, but it is not moving further because the RNA polymerase is inhibited for transcription. No enzymes are produced because simply there is no lactose in the media. Now when lactose is present in the medium, see here a beautiful mechanism takes place. When lactose is present in the medium, enzymes needed to metabolize the lactose need to be synthesized. RNA polymerase needs to be bind. All this will happen only if repressor is not allowed to bind to the operator. So lactose binds, modifies its active site. So this now cannot bind to the operator. Repressor has changed its shape, it cannot bind to the operator. RNA polymerase moves along the structural genes. These RNA will be translated into 3 enzymes which will code for the utilization of lactose. So think over it once again. When lactose is present, enzymes are needed to metabolize the lactose. Repressor protein, as it has 2 binding sites, one binding site for lactose, other for operator. Lactose, as it is present, it changes the shape of that active site to which it binds to the operator. When this occurs, operator binding site is altered and the repressor is unable to bind to the operator gene. The RNA polymerase binds to the promoter, as you saw just now in the animation. It moves along and synthesizes the mRNA, and this mRNA is produced, which is translated into enzymes involved in the metabolism of lactose. So this is what happens when there is everything is going out normally. Now there is a concept called as constitutive mutants. Means if there is a mutant repressor, what happens if there is a Lac I gene? If you create a mutation in the Lac I gene, there will be a constant synthesis of all the enzymes, even if lactose is present or not. the mutation in the lac I gene. This is called as a constitutive mutants. Now those who are studying the industrial microbiology paper, you will understand, to constantly synthesize certain products like enzymes, you can create mutations here in such regulatory genes, so that even in the presence or absence of a substrate, these enzymes will be constantly synthesized. This is called as a constitutive mutant. Okay, whether that substrate is present or not, this repressor will not be able to bind to the operator, so the polymerase will bind to the promoter, and constantly these enzymes are synthesized. So this is called as the constitutive synthesis of the enzymes. So we saw a mutant regulatory gene. Now if there is a mutation in the operator gene, I gene repressor is mutant. Now I gene is normal, there is a mutation in the operator gene. What will happen? It will not allow the binding up of a normal repressor again. The same thing happens. RNA polymerase will bind, and constitutive synthesis of the lac genes and the enzymes takes place. So this is the case where operator gene is mutant. Mutation of the operator gene or mutation of the regulatory I gene results into a state of Escherichia coli cells called as Lac-OC mutants. That is Lac-OC mutants, which are constitutive mutants, and they are always synthesizing the enzymes in the absence or presence of lactose. But remember, this is a case only during mutation. Always under a negative control, because this is repressor always blocking the operator. Now there is a positive control also. The positive control is the presence of a cyclic AMP and a CAP protein, which binds to the site of this Lac Operon. We will see that, but here let us see the positive control first. Now there is yet another form of repression, or another form of control in Lac Operon, when glucose is present or when glucose is absent. Okay, so what happens when there is a control? How does this control mechanism functions in the presence or absence of glucose? What we just saw in the previous animation was only lactose. How the lactose molecule is regulating its own biodegradation. As I said, there is another form of repression which involves the promoter region of the Lac Operon. RNA polymerase, now this is totally devoid of Lac Operon, what you just saw. Now see this as a fresh mechanism where glucose and lactose both are coexisting in the system. So RNA polymerase does not bind very efficiently to the promoter site unless there is another protein called as the catabolite activating protein, or the CAP protein. Now the CAP, it binds, CAP catabolite activator protein, this binds to the phi end of the promoter region, phi dash end of the promoter region, and at the site which is called as the CAP site, the cap binding site. So try to understand. Totally different. Now another type, it may be present in different cells, or E. coli may have both these mechanisms which we just saw right now, that is for Lac Operon negative control, or this which is called as activator control, where CAP regulation takes place. This is mostly done in the presence or absence of glucose molecules. So as I said, CAP, it binds, CAP is a protein, it is called as a catabolite activating protein. This binds at the phi prime end of the promoter, and that end is called as the cap binding site. Binding of RNA polymerase, this binding of RNA polymerase is enhanced, is increased. Therefore cap, it plays a positive control by initiating the transcription. When it is bound to the cap site, it enhances the binding of the RNA polymerase. Remember that thing. Now there is another molecule called as cyclic adenosine monophosphate, or cyclic AMP, CAMP. Now what I just said, CAP binds to the CAP binding site, but this CAP, from where does it come from, or how does it is activated by cyclic AMP protein. I will show this in the slides, but try to understand the mechanism here in animation. There is a third molecule, RNA polymerase, CAP, and a third molecule called as cyclic AMP. You have seen this molecule in many metabolic pathways. So this cyclic AMP allows the binding of this cap to the cap binding site. Means in order for the cap to bind to the promoter site, CAP should be bound with cyclic AMP. So CAP plus cyclic AMP is an activated molecule which will bind to the cap binding site of the 5' end. So in fact, CAP must first bind to CAMP, that is cyclic adenosine monophosphate or cyclic AMP protein. Then the entire complex, that is CAP plus CAMP complex, will come and bind at the site of this promoter. So this single line molecules are cyclic AMP and the cap protein. Okay, the presence, now the next question is, from where is this CAMP being produced? This CAMP or cyclic AMP is produced in the presence of glucose. If glucose is present, then this will be glucose. In general, glucose will determine whether this cyclic AMP will be produced or not. If glucose is absent, try to understand, if glucose is absent, cyclic AMP molecule concentrations are varying. How that thing happens, we will just see right now. The presence of glucose does not allow the Lac Operon to function. How does that thing take place? See here now. Now cyclic, as there is no glucose molecule, cyclic AMP levels increase. Cyclic AMP plus CAP bind to the CAP site, and polymerase is activated for transcription of structural genes, and translation takes place for synthesizing the 3 molecules of glucose. This is Lac Operon don't function. We are seeing Lac Operon in the absence of glucose. Try to understand, in the absence of glucose, cyclic AMP increases. In the absence of glucose, cyclic AMP molecule increases. How that thing takes place, that is another part of study. Means glucose is first point regulating the amount of cyclic AMP. Cyclic AMP is not present if glucose is present. If glucose is not present, cyclic AMP molecules are increasing. If cyclic AMP is increasing, then that will attach to the CAP protein. CAP and cyclic AMP complex will bind to the operator. If it binds to the operator, why this is called as a positive control? Binding will not inhibit RNA polymerase, but binding will enhance the RNA polymerase reaction, and transcription and translation will result into Lac Operon genes. All this thing is taking place when glucose is absent. Okay, so this is one point. Try to understand. Now if glucose is present, okay, if glucose is present, what happens? Glucose is present, cyclic AMP level, they are decreased, or cyclic AMP is not formed. If cyclic AMP is not formed, see, glucose is present, cyclic AMP is not produced because of glucose, then this CAP will not bind to the cap. RNA polymerase is not catalyzed for enhanced protein, it cannot bind. It will bind only if CAMP binds to this, initiating its binding. All these are interlinked to one another, and all this thing is taking place when glucose is present. Glucose present, cyclic AMP level decreased. Cyclic AMP cannot activate CAP. CAP will not bind to CAP binding site on promoter, and as it does not bind, RNA polymerase will not bind, and no mRNA, no enzymes. No enzymes when glucose is present, so automatically lactose is turned off when glucose is present. So this is what happens. There is glucose present, levels of CAMP decreases, cap cannot bind, transcription will not occur. As simple as that. So this is the activator mechanism in the Escherichia coli. So having seen this, now we will move on to see in much more detail whatever the animations we have seen. In my next lecture, I will be talking about tryptophan operon. But now come, let's see the theoretical aspects of what you have seen just now. The Lac Operon, presence of glucose, absence of glucose, inducible system, repressible system, a negative control and positive control. You have seen this in animation. I will explain it in the slides now. I hope you are understanding the things and enjoying also at the same time. So that was Lac Operon in a animated fashion. Let's try to understand it in a much more simpler fashion, and with the help of some nice figures. The whole thing will be repeated again. Whatever you just saw will be repeated again in the form of slides. So the fate of lactose as a carbon source, as you can see here, this lactose has to be taken inside. The disaccharide will be converted into monosaccharides, galactose and glucose. Here one thing which you should understand, once this lactose comes inside, its structural form changes a bit. You can see this ring structure here, and this ring structure changes and it is called as allolactose. This allolactose is basically responsible for inducing the Lac Operon. So the whole thing is lactose itself induces its own biodegradation. Lactose is taken inside by Lac permease enzyme, lactose permease enzyme, and lactose permease enzyme is a product of Lac Operon gene. So lactose as an inducer, what you just saw, let us see how it happens in the Escherichia coli cell. This is the first figure. In the chromosome or the bacterial DNA, there is a small segment called as Lac Operon, and it is always blocked by a Lac repressor. I have just shown you how this Lac repressor, which is a product of a regulatory gene, that is Lac I gene. So it is all the time, whether lactose is present or not present, mostly it is blocked at the site of the operator. Means Lac repressor is always. So therefore it is called as an inducible system. Why this is called as inducible system? Because lactose induces the operon. Lactose, its presence induces the operon system. So lactose is an inducer here. Okay, so beta galactosidase enzyme will break down this lactose into galactose and glucose. So a small amount of lactose initially is taken up and converted to allolactose, that is passive diffusion initially. This lactose, you can see 2 to 3 molecules initially, without the need for active diffusion, they are taken inside, and this lactose molecule are responsible for removing the repressor. Means lactose, it induces the removal of the repressor. Try to understand that. Means the permease enzyme which is needed, which is the last part of the gene to be produced, how is that needed first and how that is utilized is explained here. That initially few molecule of lactose, they passively diffuse inside, they remove the repressor, which triggers on the synthesis of larger amounts of the Lac Operon genes. Okay, Lac Operon proteins are synthesized. This promotes the efficient metabolism of lactose. As lactose is depleted, allolactose levels they increase, and the repressor protein, it binds to the operator site, and the most proteins involved in the lactose utilization, later on they are degraded, and suddenly there is lack of everything, and repressor, it binds to the site. So it's a cyclic process. Repressor is always bound at the operator site, and when lactose is present, it is removed. When all the lactose in the media is over, you can see in the fourth figure, all the lactose in the media is over. What happens? Even the proteins which are made, that is beta galactosidase, permease, they are degraded. Even the lactose which is present inside the cell, everything is degraded. Once lactose is over, most proteins which are degraded and lactose is over, these repressor is free to bind at the site of the operator. So this is the cluster of genes, the Lac Operon cluster of genes. We have seen this. It is made up of regulatory region, operator and promoter. Structural genes are Z, Y and A. These are the cis and trans genes, which you know very well. Lac Operon is a regulatory unit consisting of a few structural genes, and it is under the control of one promoter gene. You should know that it encodes for a polycistronic mRNA molecule. I have explained what is the concept of a polycistronic mRNA molecule, and it contains the coding sequences for 2 or more types of structural genes. Now this allows the Escherichia coli to coordinate or regulate a group of genes, that is actually 2 genes. The third is trans acetylase, which is also regulated by lactose. These two enzymes are controlled by the repressor gene, that is the Lac I gene. Lac Operon, it contains various regions, that is promoter, terminator, structural genes, operator genes, so on. And the whole idea is to put it on or put it off. Put it on when lactose is present in the medium, put it off when lactose is not present in the medium. The Lac Operon in the 20th, in just in beginning of 20th century, turn of 20th century, Jacob and Monod, they made a particular observation where they saw that a particular enzyme appears in the cell only when there is a presence of a substrate. Means only if lactose is present, these enzymes will be produced. This was an interesting observation which was known as, at that time known as, enzyme adaptation. Now this is called as Lac Operon. Francis Jacob and Jacques Monod at the Pasteur Institute of Science in Paris, they were interested in this phenomenon. At that time, Benzer's fine structure of gene was very popular. They focused on this, their attention on the lactose metabolism in E. coli, and they investigated this problem. Today, you know, lactose operon, it is a cluster of genes. The organization of this gene is in a particular fashion where you study it in two parts, that is the Lac Operon and the regulatory genes. This Lac Operon is responsible for the structural genes, and regulatory genes, these are responsible for controlling these structural genes. So two distinct different transcriptional units are there. The actual Lac Operon, which is made up of DNA elements, which contains the promoter gene which binds to the RNA polymerase, and it contains the operator gene which binds to the Lac repressor, which is a product of Lac I gene, and there is, as we saw, the activator region, the cap site, it is also present in the Lac Operon. This Lac Operon here this Lac Operon genes in this region, it binds to the catabolite activator protein or the cap protein. This Lac Operon is also made up of the DNA elements. Second part of the DNA element means the first, there are two distinct transcriptional unit. One is the actual Lac Operon, which contains the DNA elements and the structural genes. The structural genes, as I have showed you already, contains Lac Z, Y and A genes. Lac Z, it encodes for beta galactosidase, which is involved in enzymatically cleaving the lactose into galactose and glucose. It also converts the lactose into structurally different allolactose, which is isomer of lactose, and that allolactose is the one which actually binds to the repressor. Lactose directly does not bind to the repressor, allolactose binds to the repressor. So Lac Y is the second gene which encodes for beta galactosidase permease or lactose permease. It is a membrane protein required for transport of lactose and lactose-like compounds inside the cell. The last gene in the gene cluster of Lac Operon is Lac A, which encodes for trans-acetylase enzyme. It is an enzyme which modifies lactose and analogs in the preliminary study, but we will not discuss it much because we don't know its function much and it is not associated with the breakdown of lactose. The last is a Lac terminator gene here, where the whole system ends. That is the transcription termination. That is the region. Now the second part of the Lac Operon is the Lac I gene. This is the second gene, regulatory gene. We saw the first part as a Lac Operon, second part as a Lac regulatory gene, and it consists of certain genes which are not a part of the Lac Operon. They are not a part of the Lac Operon. They contain certain genes which is responsible for production of a repressor protein. It has its own promoter, that is the I promoter, and it is responsible for synthesis of a repressor. There is a constitutive production if there is a mutation in the Lac I gene. Now it encodes for a Lac repressor, and the Lac repressor, it is a protein tetramer which binds at the site of the operator. So only small amount of protein is needed to repress the Lac Operon, and it is always present. Always it is present. This tetramer polymerase repressor is always present at the operator site. Now what happens when there is no lactose in the environment? This Lac Operon is regulated by a repressor protein. When there is no lactose, this Lac repressor is active. Now Lac I, it synthesizes a small mRNA by the RNA polymerase. A repressor is synthesized which automatically immediately once it is synthesized has affinity for Lac O region, binds to the Lac O region, and it does not allow the synthesis of the structural genes. Lac Operon is therefore transcriptionally also regulated by a repressor protein. Okay, and it is also regulated by an activator protein, which we saw in the CAMP type of repression. So this method is called as an inducible method because it is induced by a lactose molecule. It is also called as a negative control because the repression mechanism is there. It is negatively regulated because it involves the involvement of a Lac repressor to stop the gene from synthesizing. And it is an inducible method because the inducer is lactose, which is converted into allolactose, and allolactose can remove this repressor. Allolactose can bind to the Lac repressor, inactivate it, and allow the transcription and translation of the genes. So this Lac Operon, which is there, is an inducible system. It is called as it is also an inducible system. Okay, why it is an example of inducible system? Because most of the time it is off. Always there is a repressor bind at the site of the operator, and it is induced when lactose is present, and lactose itself is the inducer. Lactose is the inducer. It is induced when lactose is present. Lac Operon proteins are synthesized when the lactose is present, therefore it is called as an inducible system. The main thing is that what happens always as a normal function of the gene, what happens? That it is repressed. It is bound with the repressor and it is repressed. So it cannot be called as a repressive system. It is always repressed. It is always blocked by the repressor. Now when there is lactose present, lactose will induce the system to put it on. Therefore it is a inducible system. Lac Operon is an example of inducible system. So RNA polymerase will bind to this when the allolactose, lactose is present, converts to allolactose binds. You can see there is a structural change in the repressor. This conformational change, where altered repressor can no longer bind to the operator, RNA polymerase binds to the promoter, and structural genes are transcribed and translated into the 3 enzymes which we are constantly discussing. This is one thing. Now you see the Lac Operon is also regulated by activator protein. We will see only this region, regulatory region, and the operator region, where you can see there is a cap site. This violet colored is a cap site, that is the catabolite activator protein binding site. The Lac Operon can also be regulated by an activator protein. It can be transcriptionally regulated by a second way known as a catabolite repression mechanism. And when there is a presence of lactose and glucose both, when, as you just saw in the animation, E. coli utilizes glucose first. There is no way that lactose will be utilized and then glucose. No, glucose is a simple form of sugar and glucose will be utilized first. Catabolite repression, it prevents the utilization of lactose. So when glucose is present, Lac Operon is put off. When glucose is not present, the Lac Operon will be put on. So glucose is depleted, catabolite repression is activated, and the Lac Operon genes are expressed. This sequential use of 2 sugars by the bacteria is called as the dioxic growth pattern. Dioxic growth pattern, where when there are 2 sugar molecules, the simpler one is preferred first. There may be not only glucose and galactose, there may be 2 different sugars, out of which one is simpler sugar molecule, that is utilized first, because the cell will never want to spend free energy. It will never want to waste the energy. So simple sugars are utilized first. The cyclic AMP and the CAP complex binds to the cap site near the Lac promoter, and it increases the transcription of Lac genes. Okay, as you can see, this is the cap site here, and the cyclic AMP, and this is cyclic AMP whose concentration is regulated by glucose. Okay, if glucose is present, cyclic AMP is very you can say in the presence, the concentrations are altered, and the transcription occurs when glucose is not present. When glucose is absent, cyclic AMP increases and cyclic AMP binds to CAP. Both they come and bind at CAP site near the promoter, and this enhances the transcription of RNA polymerase. Binding of RNA polymerase to the promoter is enhanced by the CAP protein. So there is a small effector molecule. The effector molecule is not glucose, it is cyclic AMP which binds and activates CAP. The form of this form of genetic regulation, it involves cyclic AMP molecule. This cyclic AMP is produced by the enzyme adenyl cyclase. Adenyl cyclase is responsible for production of cyclic AMP molecule. Now adenyl cyclase is activated in the absence of glucose. When glucose is not present, adenyl cyclase is activated which produces CAMP. CAMP binds to CAP. CAP complex binds to the promoter site CAP, and this enhances the RNA polymerase. This is a totally different mechanism, activator mechanism, and it enhances. So this is one way by which the activation in the presence and absence of glucose takes place. So lactose and cyclic AMP, if both are present, glucose is absent, this thing takes place. Lac Operon is activated by the activator protein. Now if lactose and glucose both are present but cyclic AMP is absent, lactose and glucose both are present but cyclic AMP is absent. Why cyclic AMP is absent? Because glucose is present. Now what happens? Lactose, it converts allolactose, repressor is made active. Cyclic AMP is also inactive because there is CAMP is also absent. Now transcription is inhibited by the lack of CAMP. Okay, this thing also sometimes takes place. This is inducible, and the inducible as well as the positive control of the Lac Operon. Cyclic AMP CAP complex is an example of genetic regulation that is inducible under positive control. Now here what is happening if CAMP absent? Now under mutational conditions, if this can happen and glucose is present, lactose is inactivating the repressor allolactose, and in this case the Lac Operon may be induced by the earlier mechanism which we have seen. As already seen, the cyclic AMP CAMP complex must bind to the CAP site. In the presence of glucose, enzyme adenyl cyclase is inhibited. If glucose is present, this decreases the level of cyclic AMP, so no cyclic AMP. So this cap protein is inactive. This decreases the level of CAMP, therefore CAMP is not available. Transcription rate decreases and lactose is not utilized. So if both glucose and lactose are present but cyclic AMP is absent, then there is no utilization of lactose. Try to understand. Now this is one condition where I am talking about maybe a mutation of the cyclic adenyl cyclase enzyme. If that adenyl cyclase enzyme there is a mutation, then the inhibition takes place. And what I mean to say is that even if small amounts of glucose are present and glucose is not utilized by some mechanism, like cyclic AMP is absent and glucose cannot be utilized, but glucose is present, still it does not allow the utilization of lactose. This is what is the point here. So finally let us just take a look at what is happening here. Cyclic AMP and CAMP are needed for the regulation. The repressor is always bound at the operator here. Transcription is if this cyclic AMP is present, glucose is absent, lactose is absent. If glucose and lactose both are absent, all the time this repressor will be bound. All the time cyclic AMP and CAP protein will be bound here. This is ready to enhance the binding of RNA polymerase, and this is always ready to block the movement of RNA polymerase. Okay, so and the whole thing is different when transcription is inhibited by lack of CAMP. If CAMP is absent, glucose is present, lactose is absent, then still as you can see there is no transcription or translation. These are the various types of mechanisms. So I hope you have understood the beautiful type by which regulation of gene operation, regulation takes place in the Lac Operon. When Lac Operon, how it is switched on and off. So this was the best example of a catabolite type of system where lactose is broken down. The next example is the tryptophan operon of E. coli. So this also operates at three different levels. Tryptophan operon, when tryptophan is not present, when tryptophan is present, and the third situation when tryptophan is present in low levels, low quantity of tryptophan. So this is again a beautiful, totally different perspective which we will see in the next lecture. So I wish you have you to go through this whole process once again, clarify your doubts. If you have any doubts regarding this whole enzyme mechanism and gene regulation of Lac Operon, your questions are most welcome. At the end of this lecture, down below I have given some beautiful links which will help you to understand and make notes. So follow those pathways. There is one mechanism where the whole operon system is explained in Hindi. There is one YouTube link given below where the whole operon system is explained in Hindi. You can see my lecture, go through that lecture, see all the links, take down your notes, clarify your doubts. Your questions are most welcome. But the most important thing is to enjoy these molecular level mechanisms, go to the higher level of information, become more knowledgeable and wise. All the best for that. See you soon in the next operon, that is the tryptophan operon.